Good afternoon, everyone, and welcome to Grand Rounds. My name's Tom Farden. I'm the chair and organizer of Grand Rounds. I've had a couple of weeks off. I went on summer holidays, so I've been absent from the last couple of Grand Rounds, but so grateful to those who helped organize and keep the ball rolling in my absence. This is the last Grand Rounds of the academic year. We've had a really successful year, wide range of different speakers, external speakers, internal. We've heard from medical students, we've heard from doctors, researchers, nurses, allied health professionals, and we've even heard from members of the public. So really fantastic year. This is actually the end of the 10th year in which I've been overseeing Grand Rounds. And when I took over from back in 2013, I didn't really know how long we'd be going on for. So to reach a 10-year milestone is something of a personal milestone for me. So thanks to everyone who keeps coming along to listen to these things, and hopefully we'll be here in another 10 years time, although perhaps maybe I won't. On to today. Sourav Bannerjee contacted me earlier in the year to ask for a late on slot in the Grand Round program to talk about attempting to make a new brain cancer drug. It's great, I think, to hear from our research cousins here in the medical school. So much amazing work is going on in the medical school. We don't often hear about it. I think it's fair to say that a lot of these things take a huge amount of time, and this will be the culmination of years, if not decades, of work by Sourav and those who came before him. And just to hear the output is really a fantastic thing. So I think it is a fitting end to the academic year that we hear this talk today. Usual Zoom rules apply. Please mute your microphones unless you have something to say. Use the chat function to ask any questions. There will be an opportunity to ask questions at the end. And Sourav, many thanks for coming along. Looking forward to hearing your talk.
Thanks a lot, Tom, for for allowing me to speak in the Grand Rounds. It's an honor indeed. So so my topic today is going to be my own work, which is attempting to make a new brain cancer drug. And just before I start, I would like to give you a small introduction of the background of which particular pathway, what direction this project is going to go. So. All right. So in 1992, the Nobel Prize for Physiology and Medicine was awarded to Eddie Fisher and Edwin Krebs, and for the discovery concerning reversible protein phosphorylation as a biological regulatory mechanism. So this was a groundbreaking discovery wherein they discovered that a kinase, which is a class of enzymes within our body, there's about 540 of them in total. So kinases, in fact, uses the energy currency of our body, that is the ATP, or adenosine triphosphate. They take ATP and then, it's adenosine triphosphate. There are 3 phosphate groups there. It takes off one of the phosphate groups through hydrolysis and transfers that to a substrate of interest. So a kinase has 2-pronged function. One is that it needs to bind to an ATP and then hydrolyze it and transfer it on to a target. And when a target protein gets phosphorylated, it changes its function. It could be activating, inhibiting, it could do a lot of different things. It could go down further, phosphorylate other targets in the pathway, and so on and so forth. So Eddie Fisher, who got the Nobel Prize along with Edwin Krebs, came to Dundee once, and I quote what he said. He said that in the 1970s, so Eddie Fisher was at Seattle, University of Washington, Seattle. So in the 1970s, a very ambitious and at times rebellious British guy joined my lab as a postdoc. And the first thing this British dude did when he joined my lab was to tell us that whatever we were doing was wrong. And Eddie Fisher said this, and I don't know whether you can guess, but the postdoc was actually Sir Philip Cohen. So Sir Philip Cohen was Eddie Fisher's postdoc in the 70s, and he actually helped develop a lot of different techniques in the Fisher lab, which eventually gave us more and more information of kinase biology, the physiology, and the biochemical optimization and functions of the pathways. So Philip, he was the world's most cited biochemist from 1999 to 2009. And he joined Dundee in the, I think, the mid 70s. And this is the building where he started his lab. This is the University of Dundee Department of Biochemistry. This is where he was. And then as we know it, this building now roughly looks like this, which is the School of Life Sciences. You must have seen it if you drive by Hawkill. And the MRC protein phosphorylation unit is where he basically developed this department, and he was the director for a really long time. So since Philip stepped down as director, his prodigy, if I may, Professor Dario Alessi, many of you must have heard about his work in kinase and Parkinson's. He is the current director, and from there, many of you know and love Miratul. So Miratul is also trained with Dario for the biochemical aspects. He was a clinician before. He trained as a clinical scientist in the MRC PPU, and he also focuses on different kinases in Parkinson's. And then kind of wobbling like a toddler along their footsteps is me as a newbie working on kinases in cancer. So that's kind of what my training has been with Dario, and also had a postdoc in San Diego. So largely my focus has always been kinases, phosphorylation, and developing new drugs. Right. So talking about kinases again. So as I mentioned, there are about 540 of them in total. Roughly over 25 of them has been clinically targeted, and I'll come to that in a moment. Just to remind you, the kinase has a two-pronged function. One is binding to ATP and hydrolyzing that ATP and transferring it on to a target. So roughly, all the 540 kinases look something like this. Just follow the green ribbons and the arrows there. So if you look at a kinase structure, you can see that there's one lobe, which is the smaller lobe. And then there's a slightly bigger lobe at the bottom. And in between the two lobes, there's a little bit of a gap or a cleft, and that is where the ATP binds. So as I said, almost all of the kinases have this conserved structure. They look exactly like this. If you look at their main activity site, two lobes, N lobe, we call it, and the C lobe, the bigger one, and then ATP in the middle. So if you can imagine something like the Pac-Man, where it opens up and the ATP comes in and binds to it. And so the idea was that if we had to target these kinases, that if you wanted to inhibit these enzymes in general, the best way to inhibit them would be to compete with the ATP and outcompete the ATP. So that way, it either cannot bind to ATP, or even if it does bind to ATP, and if you were targeting the same region, then you may not be able to hydrolyze the ATP. So chemicals were being made that would kind of mimic ATP structure, so that they can get into the same cleft, outcompete ATP, or if it does bind, it'll not allow the ATP to be hydrolyzed. So basically, the Pac-Man would be duped by a drug, which would look like an ATP, but would be non-hydrolyzable and would sit there in the cleft. So if you look closely, there are two aspects here. One is in the same green structure, you have the ATP, and at the same time, you have this molecule called PLX4720. This was developed by Plexicon about 10, 15 years ago. And this was a very good kinase inhibitor, which would dock right into the same clip and inhibit the activity of this kinase. Over the years, since 1999, we have about 80 different kinase inhibitors FDA approved for treatment of not just cancer. There are various other diseases as well. And the first one, sirolimus, this was not designed to be a kinase inhibitor. This was found out. It was actually discovered to be a kinase inhibitor down the road after it was approved for, I think, a rejection for organ transplant patients. It was given to avoid rejection from organ transplant patients. And then the first drug that was actually designed to be a kinase inhibitor was imatinib, and many of you may know that as Gleevec. So imatinib, just to give you a short history, was developed by a Dundee graduate in Novartis in the late 90s, and it was the first FDA-approved drug for the treatment of AML. And then I think it went on to be approved for multiple other cancers down the road. And since then, till about 2022, we have over 80 molecules that's been approved. So again, just to give you a brief examples, three very famous examples. So as you can see, I'm showing you three different kinase inhibitors, EGFR in white, BRAF, which I already showed you in green before, that was BRAF kinase. And then finally, this multicolor one is CDK4, a cyclin-dependent kinase 4. So each of these have been shown to dock with their individual inhibitors. So this EGFR, the inhibitor is gefitinib or Iressa. Many of you must have heard of Iressa. And then for this PLX4720 was vemurafenib or Zelboraf, which is no longer used, but it's one of the prime line of defense for malignant melanoma patients in the 2010s. Around that time, it is no longer used because we have better drugs for this. And then palbociclib or Ibrance is used in breast cancer patients in combination with hormone therapy or fulvestrant. And it has shown excellent efficacy as well. To give you a small idea, Iressa in non-small cell lung cancer patients in compared to standard chemotherapy, we see outstanding progression-free survival. With melanoma patients, you can see vemurafenib within 15 weeks, you can see the tumors disappearing. And then CDK4 palbociclib drug in combination with fulvestrant, you can see outstanding progression-free survival as well. So these are breakthrough drugs used extensively in the clinic worldwide for treatment of, here we talk about lung, melanoma, and breast. So what are the primary molecular mechanisms of these drugs? So when you look at molecular mechanisms, it's primarily looking at kinase cascades or the cell signaling within these cancer cells. In normal cell, all of our cells have these same pathways. All of our cells have receptors called EGFR and PDGFR. They're brothers or siblings in arms. They are receptors which are also kinases. They play a role in maintaining normal cell proliferation. So your old cells are taken away, the normal cells proliferate, they grow. But then in cancer, these pathways are hijacked. And when they're hijacked, the hijack can happen at different steps at different parts of these pathways. Each cancer has its own unique signature for these differences. So EGFR and PDGFR, although they kind of do similar things overall, but they do have distinct functions as well. They are largely pleiotropic, meaning they do a lot of different things. EGFR and PDGFR, they trigger two signaling pathways. One of them is the PI3K-AKT-mTOR. These are all kinases activating each other. So they all follow through as the same signaling pathway. And on the other side, we have the MAP kinase pathway or the BRAF-MECR pathway. Similar kinases phosphorylating each other. And the final end product is gene transcription, which would lead to large number of gene transcripts being made and then translated to proteins that will lead to higher proliferation and growth. In normal cells, it will lead to normal growth, but in cancer, it could lead to increasing the tumor size with more and more cells accumulating. So companies love these pathways. In the big industries, the big drug companies, they love these pathways. They've targeted them for years now. And some of the more newer ones from the different big pharmaceutical companies are AstraZeneca's Tagrisso or osimertinib, which was approved for lung EGFR mutated lung cancer patients. Then we have Blueprint Medicines avapritinib or Avocrit that was approved for gastrointestinal stromal tumors in 2020. Eli Lilly's abemaciclib is actually a CDK4/6 inhibitor. That's Verzenio, that's also approved for breast cancer patients. Pfizer's crenolanib doesn't have a trade name yet, so it's just crenolanib at this moment. It's ongoing for GIST as well. It's a competitor drug for avapritinib from Blueprint. Then GlaxoSmithKline, GSK's drug, very famous combination used extensively in the clinic for melanoma patients, and that's the combination of tafinlar and mekinist, or what we call dabrafenib and trametinib combinations, extensively used. And we have Novartis, whose PI3K inhibitor, Piqreg, recently got approved. And then Afinitor or mTOR inhibitor, everolimus, that hits the PI3K downstream mTOR. And then also the really old kinase inhibitors, which are approved, like Gleevec, imatinib, that was done by Nikolai Linden, Dundee's graduate. And then Tasigna, nilotinib, that was also approved for PDGFR mutated cancers. So you can see that there's a lot of activity going on in the field of kinase inhibitors, and especially targeting cancer, a lot of competitive drugs coming in, and a lot more coming out at the moment. And nearly hundreds and nearly thousands of clinical trials are going on with kinase inhibitors, either on their own as monotherapy or combination with other drugs. So this is a complicated slide, but the take-home message from this is, well, this is from 2001 when imatinib was approved down to 2021. And this is a review from Philip, which came out in 2021 in Nature Reviews Drug Discovery. This figure summarizes all the big kinase inhibitors that got approved for cancer from 2001, 2021, and they were approved for CML, GIST, AML. They were for lung, for breast, colon, some of them. There's extensively reviewed article with multiple drugs there. The problem is, when I went in and looked for those kinase inhibitors, which would specifically target brain tumors, the moment I asked the question, how many of these drugs have been approved for brain tumors, the whole figure becomes this. Not a single one of these drugs, which are exquisitely profiled, exquisite bioavailability, none of them are approved for primary brain cancers. Although you would think that it's probably brain penetrance is a problem. The blood-brain barrier is there. What if the drugs don't cross the blood-brain barrier? That's actually not the case at all. Many of these drugs are exquisitely brain penetrant, but none of them, not a single one of them, got approved for primary malignant brain tumors. So why is this the case? And that was very, very strange to me, because why are these drugs so good? They're brain penetrance as well. So why don't they show efficacy in brain tumors? So let's look at brain tumors a little bit closer then. So this is a normal brain MRI. And then there's a grade 2, grade 3 glioma, which often is resectable. Like we have neurosurgeons here like Anna and Mohammed, who I work with. They give me samples of low-grade gliomas now and then. It's relatively resectable, and you know what roughly the boundaries are, and it's more manageable. But the issue is when it becomes stage 4. In a stage 4 glioma, or what is glioblastoma. So glioblastoma is always stage 4 glioma. So glioblastoma often takes up a very strange profile. And this could be either midline invasive, or the issue, my primary issue is you often do not know what the boundaries are. So there are ways in doing surgery to try and understand what's the normal brain and what's the malignant brain tumor. You often are given a pink drink that allows, and that goes to the brain, makes your tumors a bit more brighter to look at under fluorescence. You can take the tumor cells out, but you almost never get the whole thing out. And that is the biggest problem. And hence this particular tumor, or glioblastoma, is often referred to what is a gum on carpet scenario, meaning that if you put a chewed gum on a carpet and try to take it out again, you're never going to be able to take the whole thing out. Some of the gum will still remain on the carpet. And that's exactly what happens. Those remainder gums gives rise to a whole new gum. And that's why glioblastoma is so difficult to treat, to take out. So they're almost always refractory. And in many cases, a headache is often too late. If a person is having a headache, it's probably already stage 4, and it's already, there's a huge tumor there. So they often do not manifest early enough for you to catch them at lower grades. So, okay, so that's the tumor. So what's the problem in the molecular mechanism? So why can't we target it? Again, there's a huge distinct difference between how the brain tumors are looked at by us academics and the pathologists. And well, it's not something that we disagree upon. It's just a different way of looking at it. In a pathology, you're looking at an endpoint, right? So you have the tumor on a block, you look at the pathology, that's the endpoint tumor. And the endpoint tumor, the pathologist will tell you, like Colin Smith in Edinburgh, the brain bank will tell you, well, there's oligodendroglioma, there's astrocytoma, and then there's diffuse midline. And if I were to ask any very learned audience, even this audience here, if I were to ask the question, when I say oligodendroglioma, what do you think the cell line of origin is in the brain? I'm pretty sure many of you would say it's oligodendrocytes. And then if I ask, what about astrocytoma? What is the cell of origin for astrocytoma? And you would invariably tell me that it's astrocytes, right? And that's obvious. That's what the names are. But in fact, glioblastoma, or the DMG especially, do not grow from adult differentiated oligodendrocytes or astrocytes. The tumor shows more positive staining for oligodendroglial markers and astrocytic markers in the tumor, but it does not mean the cell of origin of astrocytes and oligodendrocytes. In fact, if you look at the brain cells, we all know there's a neuron, we know there are dendrocytes, oligodendrocytes, there's microglia, and there's astrocytes. But actually, there are two more population of cells in the brain. And those two are the oligodendroglial progenitors and the neural progenitors. The progenitor cells are the brain stem cells. And glioblastoma, almost 100% of the time, arises from the progenitor cells of the brain and not the adult cells. Because it's growing from the progenitor cells, glioblastoma is a stem cell disease and not just an epithelial lining disease or a ductal carcinoma or something like that. It is a stem cell issue. And you know what stem cells do. Stem cells have the ability to change themselves. So an oligodendroglial progenitor can give you oligodendrocytes, can give you astrocytes, can even give you neurons. NPCs can give you astrocytes, can give you neurons. So they can change, they can differentiate. They give you different types of cell populations. And recent studies have shown that a single brain glioblastoma tumor, one tumor from one patient, can have 4 distinct stem cell populations in that tumor. And each of these distinct stem cell populations expresses distinct oncogenes, distinct markers. Like this particular green population here expresses PDGFR, which we discussed a little bit. Then this yellowish population expresses EGFR. And then we have this pink red area, which is just anaplastic, meaning they don't resemble any cell type in the brain. They just are completely messed up. Okay? So if you're going to target with a specific PDGFR molecule like avapritinib, or if you're going to target with the Tagrisso or EGFR drug, you're only going to hit one small population of the brain tumor, while the others survive and give you refraction and come back. That is the biggest issue. So if cancers are X-Men, glioma is the mystique of the tumor. So now when this was really strange for me, so I started looking at what makes brain tumors so different. So firstly, I looked at what are the molecular pathways that's generally driving brain tumors from other people's work and other publications. Unfortunately, nothing new cropped up when it came to proliferation of the tumor. It was the exact same players. It was the same EGFR, PDGFR, the same MAP kinase pathway, same PI3K/AKT/mTOR, so it was still the same. So at this point, a lot more literature started coming up, especially in the last 5 years, saying that targeting just the proliferation through these pathways are not enough. That is because these cells are not normal cells, these cells are stem cells. And so, of course, we started applying stem cell biology to them, like normal stem cell, hematopoietic stem cells, brain stem cells, the normal stem cells in the system. We asked the question, what is the main pathway that drives stem cells? And the pathway that drives stem cells is called a Wnt signaling pathway. And this Wnt signaling pathway is in fact distinct from the PI3K/AKT/mTOR or the MAP kinase pathway. So when I looked at the Wnt pathway, I started looking for kinases because that's what I do. I work on kinases. So we saw that there was this group of kinases called DYRK and CLIX, which were playing some sort of a role in the Wnt pathway. And by some sort of role, I mean activating the Wnt pathway. And so my thought was, can we actually target the DYRK and CLIX? This hasn't been done clinically before. There are no clinical DYRK/CLIX inhibitors reported. Lots of molecules have been made to target DYRK/CLIX academically, but not really taken to the clinic per se. So I looked closer to these kinase families. So I found out there were like 5 different DYRK enzymes, kinases, and 4 different CLICs. So we kind of need to target 9 kinases at the same time to be able to hit all of them to make sure the Wnt pathway does not progress. So yeah, it was an uphill task. So it was also sounded crazy. We had to hit multiple targets. Instead of making a molecule more specific, we had to backtrack and make it less specific, which was interesting. So yeah, I reached out to a few chemists, and then I met this person, Chris Hume, who's a professor at University of Arizona. He's an outstanding chemist. And his MO was that he was targeting Alzheimer's disease, okay? And what he discovered was a DYRK molecule, not DYRK/CLIX, just the DYRK. If you just inhibit the DYRK kinases in an Alzheimer's animal model, you reduce the tau aggregates in the brain. And from there, he currently has a molecule that's in preclinical development for Alzheimer's disease. And it's a very specific DYRK inhibitor, not a very broad spectrum one. It's just specific for DERK. But one thing he has completely nailed is brain penetrance. His molecule is 90% brain penetrant. So he's got the brain penetrance nailed. He's got a very good drug that's gonna go into Alzheimer's drug. So I asked him a question: can you backtrack and give me something less specific? Okay. A very strange question to ask to a chemist. Can you backtrack and give me something less specific? So yeah, so the plan was, it was a crazy plan at the time. We have to hit DERK clicks. The same molecule has to hit PDGFRA. And then initially we thought we want to hit EGFR as well, but that we know from previous work that it gets toxic in the brain. You get brain inflammation if you hit EGFR because you kind of need that for normal brain development as well. So we decided to hit the PI3K instead, one downstream. So our molecule, hypothetical molecule, has to be brain penetrant, has to hit the DERK clicks, has to hit the PDGFRA, and has to hit PI3K. That's the challenge. But only that way we'll be able to hit both the survival pathways of cancer and the stemness, reciprocity, plasticity, refraction, the stemness that has to be targeted. Because if you cannot hit the stemness of glioma, the population is going to keep coming back. So if we hit the stemness, we hit the proliferation. 3 birds with a stone. So this is the plan. So we take Chris Hume's DERK click. Then we take Blueprint's Avocrit, which is a brilliant PDGFR inhibitor approved for GIST. And then we have Novartis's Piqray, which is a PI3K approved for breast cancer. And the idea was, if Chris Hume, both of us sit down, I'm the biologist, is the chemist, we discuss and see if we can combine the best bits of the 3. We keep the brain penetrance, keep the DERK click activity, add on Blueprint's Avocrit molecules, functional groups that would hit PDGFR, and put in Piqray's, Novartis's Piqray's molecules, parts of it that makes it PI3K. So we put together sugar, spice, everything nice, and from the cauldron jumped out something weird. Okay. So this molecule, again, cutting a long story short, DYR726. This molecule, if you just focus on the bottom 3 factors, so we have a half-life of 8 hours, its oral bioavailability is 57%, and its brain to plasma ratio 99%. So it's 99% brain penetrant. But then again, this is a very artificially developed, very a crazy chemistry molecule. So we wanted to make sure that this was safe even before we started it. So to check whether this molecule was safe, we decided to treat normal colon epithelial cells. And when we treat normal colon epithelial cells, you can see the cells are not really dying. So the cells were flatlining, there was no death whatsoever, and the cells were roughly all alive. So this didn't really kill normal epithelial cells. And then we actually treated a mouse with 9 doses of 75 mg per meter square. Roughly the human dose is 75 mg per meter square over 2 weeks. The mice were perfectly fine, no problem at all. So we decided to, now we are currently testing a daily dose of 75 mg per meter square to see if the mice show any sort of toxicity, anything like that, because this is an experimental molecule. So we want to make sure it's safe. And yeah, for now, it looks like it's pretty safe. So we decided to test whether this had any specific capacity to kill glioma cells, which is what we want to target, right? We want to kill the glioma cells, the cancer cells. So this is a brain tumor growing on a dish, okay? So this is the primary patient-derived tumor cells that we have. We put them in a dish surrounded by a matrix, which kind of resembles the brain. It has like collagen and a few other matrix proteins around it. So you make this 3D culture, it grows inside the matrigel, and then you can see, just like a brain tumor does, it starts spreading. The cells come out of it and it starts invading the surrounding tissue. It spreads and goes up. The moment we add in low dose of our drug, 726, you can see there's a complete collapse. All the cells which are spreading out and trying to invade into the matrix, there's a collapse and form a ball of cells. So it did not allow the tumor to spread. It just holds it in place. So okay, it can hold it in its place. Can it kill? Can it kill the tumor? Can it get rid of the tumor? So yeah, so this is the same tumor that was not in a matrigel, so we're not allowing it to grow too much. We just took the tumor cells as a 3D. We treated them with the drug over different concentrations, and you can see by about 1 micromolar, we saw majority of the tumor cells gone. This is one example that we really had to search for. And then by 3, there's hardly anything there, and by 10, they're almost all gone. And then we saw that both in formed 3D spheres, and also before they formed a 3D, we still treated them to see if they can form 3D. And indeed, they really cannot really form 3D at all when you have the drug in the presence. So remember, they didn't kill any normal colon cells. It was only targeting the glioma cells. And of course, as good scientists do, we quantified across multiple replicates, we carried out statistics, and you can see that by 1 micromolar, you see significant reduction in glioma cell population, 3D population over time. So it does 2 things. It hits them, it kills the cells over time, and also stops it from spreading. So you can do both. So next, we wanted to benchmark this molecule against some of the very big names in the field, like Tasigna, Novartis Tasigna, Gleevec from Novartis, and also Avicret, that is avapritinib's drug. And the best drug that's currently thought to come out in market is still experimental. It's still in clinical phases, and that's crenolanib from Pfizer. So you can, this is a cell death data. So you can see more concentration of the drug, zero drug, 0.3 micromolar, 0.5 or 1 micromolar as you increase the drug concentration. Crenolanib is the best drug currently, Pfizer's drug, and then the others also. Gleevec doesn't really do much, Tasigna a little bit, avapritinib does a little bit, Avicret does a little, but then when you look at 726, our drug, you would see that by 0.3, it completely crashes the primary glioma line. So this is a patient who had a very high PDGFR amplification in his tumor. So we got the tumor cells and then used the primary dissociates from the tumor. And you can see 726 seems to be better than crenolanib, Pfizer's drug, Novartis's drugs, and also Blueprint's currently that's undergoing clinical trial for brain cancer. So it's benchmarked really well. And then more recently, the combination, the skin cancer combination of Tafinlar and Mekinist, that is dabrafenib and trametinib, got approved for a very small population of brain tumor patients. So both of these drugs are brain penetrant. And for a very small population of brain tumors where they have a specific marker called BRAF V600D, this is more seen in skin cancer patients, where some brain tumor patients do show the same mutation. And in that, there was a very small trial done. It's in Lancet Oncology, where there were literally 50 patients in this clinical trial because it's a very rare mutation in brain tumors. So they used a combination of Tafinlar and Mekinist and clearly showed that they significantly improved the quality of life for these patients with the V600E mutation marker. So I wanted to ask a question. Does 726, our drug, able to target the same V600E tumor? So the Mayo Clinic had a patient whose tumor was collected, and this tumor, GBM44, this patient's tumor had V600E. We got the cells, and then we used a clinical combination on the cells of both Mekinist and Tafinlar, a combination. You can see the death in orange, the combination death. And then 3 micromolar of 726 alone is pretty much similar to the combination of the 2 drugs. So it seems like 726 could probably be more of an umbrella drug for many of the different pathologies seen in glioma. So that might be a good news, but of course we only have 1 patient sample. It's a very rare tumor. So we only have 1 patient with sample with this particular mutation. Hopefully we'll get more from the Mayo or from our neurosurgery team here, and then we'll be able to take it forward. So finally, a few specific cases that we've done. So there are many atypical glioma. So glioma is not one mutation. There's multiple different mutations that can take this. So the green line is 726, and we're combining it with Novartis, dabrafenib, osimertinib, Tagrisso, with all the different drugs that's currently undergoing clinical trials for glioma as well. So just like you can see here, buparlisib is actually a PI3K drug from Novartis. And you can see it's pretty similar to our drug. And then this particular patient's GBM10 actually probably went through a Tagrisso clinical trial from AstraZeneca. So that's osimertinib. It's also called Tagrisso. So you can see in orange, that's Tagrisso, whereas 726 is much more potent than Tagrisso. Similarly, this is an Afinitor. This drug is Afinitor, an mTOR inhibitor, Afinitor. And you can see Afinitor doesn't really kill the cells at all, whereas our drug was able to kill the same tumor cells pretty prolifically, no problem at all. And then finally, we have this DIPG line. DIPG is a diffuse intrinsic pontine glioma. So this is pediatric. So if, I mean, from my own opinion, DIPG is one of the worst cancers that there is because the survival rate in kids is extremely poor, less than a year. The surgery is very difficult. Radiotherapy also doesn't really improve the livelihood by much, and hardly any drugs are available whatsoever. So we got this DIPG line from ICR in London. And from there, we got the tumor, we got the cells, and we tested 726 and a derivative of 726. We're also working to make it better to 747. And you can see that it's killing the DIPG cells pretty prolifically. So that's actually a very good sign. We're probably going to expand into pediatric glioma as well with this particular drug. So just to wind up, so this is the first in class. So I do not think anyone else has made a trifecta PI3K PDGFR Wnt pathway inhibitor. It's orally bioavailable, 57% orally bioavailable. We have to improve that a little bit more. 8-hour half-life, that also needs a little bit more improvement. We can try and see if we can make that better. We are expanding into pediatric DIPGs, pediatric diffuse intrinsic pontine glioma tumors, and we have started in vivo experiments. So this is the model that we use. We take a mouse, we cut open the skin, make a small burr hole in the skull, and then we inject primary patient tumor cells. And then we actually have luciferase in there so we can track the tumor growth using imaging, live imaging. We don't have to open the skull, we can see the image straight up, and you can see the tumor progressing. And once the tumor reaches a particular threshold, we're going to start treatment and then track and see if the mice survive better. So we can do an overall survival and also progression-free survival in mouse models before we think of taking this further anywhere near preclinical or clinical development. So yeah, so of course, this is not just me doing the work. There's a lots of people involved. The laboratory, we have Vasudha, Amy, who are currently PhD students in the lab, and then Kina, the research tech. It's primarily Vasudha's PhD project, this particular operation. And Kina really helps with that as well. As you can see, it's a huge project in general. We recently joined by Ira, who's a postdoc, and Lyndon. Many of you might know Lyndon. He just got the Dean's Award. So Lyndon worked with us for quite a bit as a master's student, and then Joy, who recently joined as a master's student, and then Aditi also is contributing to the same project as an undergraduate project student. And of course, we have a lot of different people involved. We have biochemists, then Chris Hume, who's the chemist. DDU down the road helps us with the drug development and DMPK studies. We have Anthony Chalmers in Glasgow and Hannah Lord here in NHS, and also Russell Petty, who's kind of advising me along the way. We have Anna and Mohammed in the NHS Neurosurgery, helps me get set up a biorepository here at Dundee for the first time, and we are collecting patient tumor samples, making them into cell lines as well. And of course, funding, nothing is possible without money. So we recently got funded for this project, about 1.75 million from the Critical Path Institute in the US. And then we have Mayo Clinic providing us with more optimized and genetically sequenced tumor samples. And then 10 of us, UKRA Future Leaders Fellowship, NIH funds Chris Hume's lab, and then Nine Wells Cancer Campaign got me going when I started in the lab in 2020. And I'm happy to take any questions, comments, or if you want to reach out later on for any other queries, you're more than welcome. And thanks, Tom, again for the invitation.
Thanks, Sourav. I said this was a culmination of a lot of work, but as you demonstrated at the beginning, standing on the shoulders of giants here. Yeah. I think you put yourself down somewhat as saying you're the newbie in the department. You're doing some amazing work here, absolutely incredible stuff, and we're really grateful that you're prepared to present this work, which is at an early but pivotal stage. I mean, it's really fantastic stuff. Now, has anybody got any questions? There's no question in the chat, but has anybody got any questions? I'll just try and change my screen to look at the. No one has their hand up currently.
Anybody can reach me out by email if you have any questions later on. No
so
problem.
My colleague Dave Connell shares my office, so he's going to just tilt my screen a little. There you go.
Okay.
Hi there, that was inspirational. So thank you so much for bringing that to Grand Rounds. And I guess it's just it's quite a generic question for those of us who are interested in clinical academic work. Let's say that the mouse model work is successful. What would be your next step in terms of bringing this into clinical trials? Just to be fascinated.
Right. So yes, that was a question for me as well when I asked Russell Petty what would be the next step, because I'm a preclinical, I'm a kinase person, right? A biochemist who kind of got into clinical drugs and developing them. So the main step would be getting the cGMP synthesis done. If it works in animal models, and then there's a few other small hoops to jump, like toxicity studies in 2 species tox, like you want a rat and then potentially another tox in species, and then cGMP synthesis. So we have to make this properly clinical grade synthesis done. That's a kilogram synthesis that costs $1 million. Okay, so that's a lot of money. And that's where generally we either have a patent, we already have a patent on it, we're probably going to give it to a company or start a startup. I mean, that's going to come once the animal model's done. So 2 species tox, and then from there, GMP synthesis. And from there, if everything's done, then we can start a phase 0, like you recruit 5 or 6 patients, something we discussed with Russell Petty and Anthony Chalmers. 5 or 6 patients of any tumor, any solid tumor, and then see the efficacy within that. So that's generally how this would progress.
And out of interest, how long does that take? The 2 species toxin and the GMP, is it like a year, 2 years, or?
It's probably like cGMP is all about money, unfortunately. We have to get £1 million from somewhere to make a cGMP synthesis. That's doable if we have the animal model working brilliantly. We already can see some effects on animal models, but it's still too early. We just started it. So we'll know more maybe in a month or 2. But yeah, I mean, it varies a little bit. We have to see how to do the 2 animal talks, and once that's done, whether it's a month thing or a 6 months thing, it needs to be looked. It's too early for that, actually.
Thanks so much.
I'll swizzle you around to my ugly face. There we go. Glenn, no second name, has said, is your pleiotropic compound approach applicable to any other cancers which have poor treatment outcomes?
Yes, it is. There are ways so there are a few specific targets. Like, for example, when you're hitting a kinase, you're never going to hit only that one kinase. You often have a few off targets, but you can control that off targets. We have actually kept 2 off targets, for this particular molecule, and those are hitting KIT and FLIT. So KIT and FLIT are amplified in hematological and GIST. So the idea is, if for any reason glioma angle doesn't work, we can easily repurpose this molecule into breast because it's hitting PI3K, GIST because it's hitting PDGFR. We can hit FLIT, we can hit AML and CML because it's hitting FLIT. So yes, we can repurpose this for any tumors if it is efficacious there. Like, for example, if it's a tumor which is really out there and none of the mutations are there that I was talking about, of course the drug won't work, but I'm pretty sure on a more general scale we can. Yeah, that's about precision medicine, I'll have to say. Yeah. We can definitely take it forward to other cancers, no problem. But we are starting with the most difficult one.
Yeah, absolutely. And you are starting with a really difficult one.
Yeah, it's probably the most difficult one.
I don't do really any cancer work beyond the acute presentations on the ward, but I did do some cancer work in my training, obviously, in early consultancy, and I remember very clearly gefitinib and then erlotinib in lung cancer, and how limited we were in terms of use of it, and how it was very exciting, but early stages. And now you look at that diagram you have of all the different kinase inhibitors, and I mean, just a massive explosion that I am so much time for my oncology colleagues who, their practice is changing on a year-to-year basis. In fact, perhaps more frequent than that. And that's and just you've been talking about how long it's taken to get here. And Dave's question says, there's years and years. If this were to go absolutely perfect, to get this to market is still 5 years plus easily.
Yeah, easily. I mean, I'm not even thinking about market right now. It's just, I'm just taking one step at a time because the chances of failure are very high. I mean, at this stage.
And that, and yet you bat on, which is amazing.
Oh, absolutely. Oh, yes, that's what I'm here for.
Well, that's it. As Dave said, it's an inspiring talk and a really fantastic way to end this academic year's Grand Rounds. I hope you consider coming back, give us an update.
Sure.
Interested to hear what your PhD student's doing, and the rest of the lab if you've got any other work. But this is fascinating stuff, so please do consider coming back to Grand Rounds. Thank you very much.
Thank you very much. Thank you very much for having me. Yeah.
Oh, it's our pleasure. To everybody else in the audience who's still here before nipping off to clinic, thanks very much for attending. Thanks for your attendance all the way through this year. Grand Rounds only happens because people come along to it, and I think that the ability to do it now over Zoom and over screens has certainly helped matters. But I'm grateful for you still signing in every week and coming to watch and taking part. And I'm grateful for all the people who volunteer to give their talks. Now, if you want to give me 1 second, I just need to share my QR code. If you want to get recognition that you've been here today, then you can scan this QR code that's on your screen. That will then take you to the registration page where you can fill in the details of today's Grand Round. You'll be emailed a certificate, and then we keep a record of who comes to Grand Rounds. Please do offer us feedback if you think the program is good, bad or ugly. Even better, please volunteer to do a talk in the next academic year, which will start sometimes in September. Again, thank you to Sourav, thank you to everybody who's spoken at Grand Rounds and anyone who stood in for me when I've not been able to make it. And I look forward to a good summer and seeing you again in the next academic year come September. Thanks very much, everyone. Enjoy this lovely patch of weather. Have a great weekend, and I'll see you around.
Thank you.